Hello friends, today I'll be discussing the renal physiology in relevance to the diuretic action. So, the basic functional unit of the kidney is the nephron, and I have made the structure of a nephron over here. So, this nephron is it serves the purpose of filtration, reabsorption and secretion, and these three functions they help in formation of what we call as urine. So these three processes I'll be discussing as I go along, and this will make a background of what we discuss in the next segments, that is the diuretics. So starting with a little bit about the structure of this nephron. To begin with, there is one Bowman's capsule over here. This structure, this is called as a Bowman's capsule, and in it there is presence of a tuft of capillaries, what we call as the glomerular capillaries, and this capillary is supplied by one arteriole, what we call as the afferent arteriole, which brings blood to this, and then there is another one what we call as the efferent arteriole, which takes the blood away from the glomerular capillary. And this region here, the glomerulus, it as such acts as the filtration unit. So whatever blood comes over here through the afferent arteriole, it is filtered across the membrane of the capillary and across the membrane of this Bowman capsule into this region, into this tubule. So that is that is how filtration takes place, and this filtration depends on some forces what we call as the Starling forces, and these Starling forces are capillary hydrostatic pressure inside this glomerulus and the capillary hydrostatic pressure outside the glomerulus, that is in the Bowman's capsule. Then osmotic pressure exerted by the colloids inside the blood, as well as the osmotic pressure which is provided by the colloids outside this. Now this filtration occurs through pores. There are many filtration slits inside this region in this capillary. So simply stated, most of the electrolytes, they are filtered through this, but the larger molecules like plasma proteins, they cannot be filtered, and as well as the blood cells, they are not filtered. So and you know that the osmotic pressure is mostly contributed by the plasma proteins. So since there are no plasma proteins coming out into the Bowman's capsule, so there is literally there is no osmotic pressure exerted from the outside. Okay, so osmotic pressure normally tends to retain the water. Normally wherever the the colloids are high, they will not allow water to get out of that compartment. So if there are more of plasma proteins inside the blood, they will not allow water to move out, to filter out. Okay, but the capillary hydrostatic pressure, the more it is, it will lead to more filtration. Okay, so these are the forces which are responsible for filtration, and as I have told you, most of the electrolytes, they are filtered, and the plasma proteins as well as the red blood cells, they or the blood cells as such, they are not filtered out of the blood. So what we get here is a simple plasma minus plasma proteins. Okay, that is what we have in the Bowman's capsule, and the total glomerular filtration rate is 125 milliliters per minute, which amounts to 180 liters per day. And you know that the plasma volume is only about 3.5 liter, so you can say that the whole blood, it is filtered about 60 times in a day. So that means it is a very efficient process for filtering everything which is present in the blood, including all the metabolic end products, which is the actually purpose of this filtration, so that those substances, they can be filtered out. So filtration follows simple principles. Molecules, the size of the molecule, it has to be small. Smaller molecules, they pass easily, and larger molecules, they stay back. So, and one more thing is the substances which are bound to the plasma proteins, for example drugs, so they are also not filtered because uh, they are bound to the plasma protein, and plasma proteins are not filtered. So any drug which is bound to plasma proteins will not be filtered through this process. There are other methods for excretion of those substances. Okay, anyways, now when it has been filtered and the blood, the plasma, it reaches this, or you can say the filtrate, it reaches this Bowman's capsule, it moves to the next segment, what we call as the proximal convoluted tubule, or the PCT. And in the PCT, what happens is, there is this PCT actually handles most of the filtrate uh, which has been filtered. The thing is, as I am telling you that 180 liters is whatever is filtered from this glomerulus. So you know that the urine output is not more than 1 or 1.5 liters. So most of whatever is filtered is reabsorbed, and that reabsorption occurs in these segments, as I tell you. So proximal convoluted tubule, it handles, or you can say it reabsorbs 65% of the filtered water, 65% of the filtered sodium, nearly 85% of the bicarbonate, and virtually all of the amino acids and glucose. So all these things, they are reabsorbed in the proximal convoluted tubule. Okay, and and one more important thing is, here the reabsorption is isotonic, means as many ions are moving out of this PCT, uh, the same amount of water also moves along, since this segment is freely permeable to water. So the isotonicity is maintained. So the same osmolarity which was present in the plasma, or which was present in the Bowman capsule, it is maintained in the PCT only, in the PCT. So when it passes next, so what is the condition here? It is having the same osmolarity, okay, and another thing is 65% of sodium has been reabsorbed, most of the amino acids and glucose, they have been reabsorbed, a large fraction of bicarbonate has also been reabsorbed. Now what are the actual processes, that I will be telling in a later section. I am just giving an overview. Now till this point everything is isotonic. Okay, now what happens is, then, and one more thing is that this PCT, it lies in the cortical region, or the cortex. The kidney can be divided into two parts. One is the cortex, which is on the outer side, and the medulla, which is on the inner side. And now it moves into this tubule, it moves into the medullary region, and in the medullary region it also changes its shape, its structure, and that structure is what? It becomes thin. So it becomes very thin. The lining cells change from cuboidal to squamous, and then it forms what we call as a thin descending loop of Henle. This whole arrangement, this loop is called as the loop of Henle, and this part which is going down into the medulla, it is called as the thin descending loop of Henle. Okay, then it goes down into the medulla, then it takes a U-turn and goes up, and it starts ascending as the thin ascending loop of Henle. Okay, then in the medulla only, it changes its shape again into what we call as the thick ascending loop of Henle. So this is the thick ascending loop of Henle. This thick ascending loop of Henle continues into the cortex, goes into the cortex, and then it again changes its shape and structure, and obviously the cell type. and it is converted into what we call as the distal convoluted tubule, or what you can call it as a DCT, a distal convoluted tubule. And it is important to mention here that this DCT, it moves in close proximity to the same Bowman's capsule. Okay, and this is responsible for one feedback mechanism, what we call as TGF, or tubuloglomerular feedbacks. So that will be discussed later on. Now this DCT then converts into what we call as the collecting tubule, moves next into the collecting tubule, and finally it goes into what we call as the collecting duct. And these collecting ducts, then so many collecting ducts all over the kidney, they get together, come together, and they open into the renal pelvis, from where the urine is moved out, and it goes into the ureter and finally excreted. So this is a general outline about this. So I had discussed about the proximal convoluted tubule, what it is doing. Now coming to the thin descending loop of Henle. This segment is freely permeable to water. And there is one more thing, that this region, the medulla, why it is different is because the medullary region has got hyperosmolarity, means the osmolarity here is much higher than it is normally found. So normally the osmolarity is to the tune of 300 milliosmoles. So in the medulla you can find up to 1200 milliosmoles. So there is a lot of extra electrolytes lying there and less of water. That's why it becomes more concentrated. And since the thin descending limb is freely permeable to water, you can expect that water will move out of this segment into the medullary interstitium. So there is movement of water inside, and when there is movement of water inside, what will happen to this? It will become hyperosmolar. The tubular fluid inside the loop of Henle, while it is descending down, it becomes more and more hyperosmolar, because the water has been removed and the electrolytes stay inside because there are no channels for their movement. So the osmolarity around this region is the same as that of the medullary interstitium, so they kind of equilibrate. Then what happens is, moving further, when it reaches the thick ascending limb of loop of Henle, what happens is, this region now becomes impermeable to water. Now becoming impermeable to water means what? There will be no exchange of water to the outside now. Now since there is no, but there are channels which are operational here and which are responsible for extracting various electrolytes from this tubular fluid. So what happens is, at this region, I'll mention one channel. There is one channel which we call as sodium potassium 2 chloride channel. So there is one sodium potassium 2 chloride channel which is functional in the thick ascending loop of Henle, and what it does is, it causes movement of these 4 ions. So sodium, potassium and 2 chloride. You know, sodium is a positive charge, potassium also has a positive charge, and chloride has a negative charge. So since there are 2 chloride, this movement is electronically neutral. Electrically this movement is neutral. So these 4 ions are removed from the tubular fluid here, and since there is no net movement of water, what will happen? This will keep on moving and the water inside will be trapped. So what will happen? There will be dilution of this tubular fluid in the thick ascending loop of Henle, and that's why this region is called as a diluting segment, and it handles nearly 25% of the tubular load of sodium is handled around in this region. I told you 65% is dealt with in the PCT, 25% is dealt with in the loop of Henle. And another thing that happens is that the osmolarity of the fluid, of the tubular fluid, keeps on decreasing. It becomes hypoosmolar. Okay, Then coming next, there is another segment what we call as the DCT or distal convoluted tubule. In this DCT, what happens is, again there are channels which cause the movement of ions, and they cause a movement of one sodium and one chloride, or you can say salt as such. So movement of salt outside from the tubular fluid, what does it cause? It causes further dilution of the tubular fluid. So this thick ascending limb of loop of Henle and the distal convoluted tubule, they are combinedly they are called as the diluting segment of the nephron. So as you can expect, reaching this point, what will be the scenario? The tubular fluid will be highly hypoosmolar because it has been diluted. Much of the electrolytes, they have been taken away from it, but water has not been allowed to move. So this segment here, the osmolarity goes to a very low level. Okay, then coming to the collecting tubules and the collecting duct. Here the absorption of sodium depends on the presence of mineralocorticoid, and the mineralocorticoid which is responsible is aldosterone. And aldosterone, what it does is, it causes reabsorption of sodium in exchange for potassium ions. So you can say nearly one sodium ion is reabsorbed and one potassium is replaced in its place. So there is a net sodium reabsorption, but at the same time there is potassium excretion, and this process depends on aldosterone. If there is no aldosterone, what will happen? The sodium will stay in the urine, it will be excreted in the urine, while there will be no secretion of potassium ions. And not only potassium ion, there are also one type of cells what we call as the intercalated cells, and they also secrete hydrogen in exchange for sodium at this point, and that helps to regulate the acid-base balance of the body. So that will be discussed when I am discussing the classes of diuretics. And then coming ultimately to this collecting ducts. In the collecting ducts, what happens is, now you can imagine there is only a hypoosmolar fluid which is moving in the collecting ducts, and here this segment is again impermeable to water under normal circumstances. So there will be no movement of water outside of this. So if this is the situation, what will happen? A hypoosmolar urine will be passed. But there is one hormone called as ADH, or what we call as antidiuretic hormone, or you can call it as a vasopressin. What it does is, it comes into play in this segment, and what it does here is, it causes formation of one pores for movement of water, what we call as aquaporins. So these pores, what they do is, I have already told you that in the medullary region the osmolarity is on the higher side. So water will have a tendency to move out, but there are no pores. So whenever you give ADH, what will happen is, these pores will open up, and then water will move in direction of its concentration gradient from a dilute to a more concentrated solution. So it will go out, and ultimate result will be, there will be formation of what we call as a concentrated urine, because all the water, it would have been absorbed into the medulla, and ultimately a concentrated urine will be formed. So in absence of ADH, a dilute urine will be formed. So this is a little bit about the physiology of urine formation. Then I will be taking up more things when I am taking up each class of diuretics. Then there is one more very important factor, and that is how this osmolarity of urine is of this medullary segment is formed. That is something important to discuss. So as I told you, there is a sodium potassium 2 chloride channel here in the thick ascending loop of Henle. Whenever it reabsorbs this sodium from the tubular fluid, where does it go? all these things, they go into the medullary interstitium. So they add to the pool of electrolytes here in the medullary interstitium. So this is what contributes to the increase in the medullary hyperosmolarity. And this mechanism, due to this mechanism, the loop of Henle serves the purpose of what we call as a counter current multiplier. So due to this counter current, it keeps on becoming more concentrated. It provides more concentrated you can say filtrate here, and that electrolytes are taken up in a large amount from this into the medullary interstitium, which further contributes to further concentration of the tubular fluid. So it keeps on repeating itself, and ultimately this level is reached, nearly 1200 milliosmoles. So that is how that is the basics of renal physiology with relevance to pharmacology. Then I'll come to the various diuretics, which I'll be taking up in the next sections. Thank you so much.